Well, thank you very much for this great honor, and thank you very much for allowing me to be here. It's really a fantastic conference, Alejandro. It's really, I've been enjoying it a lot. I've seen a lot of different biology, which we learned this morning is very good for the night science part of the brain, so I'm very much happy with this. Let's see, where did I put the electronics? Yeah, so this is actually a lecture which is made possible by EMBO. So I'll tell you a little bit about EMBO, I promised, to the EMBO Society. It's an organization in Europe that tries to really, an ambassador for molecular biology in Europe. It's not a major funding agency, although what's really important is that they give out fellowships for young investigators, for people that want to be postdocs, and really allow people all over the world to come to Europe and do research. So if you're interested and you want to come to Europe, don't forget to check out the EMBO fellowships. I already saw a few of these EMBO fellowships in the acknowledgement of people, so it's really a great way to be funded. Now we have to switch to my presentation. And actually, I have been working on the presentation all week. I decided, I thought I would talk about something else, but then I was so inspired by some of the talks that were related to translation that I really focused the talk on translation and detecting translation in individual cells. Now, our lab gets really excited if we can measure something that was never measured before. That's kind of what we're going for. And we have been doing that in single cells since I started the lab in 2000 at MIT, and also we're doing it right now in the Netherlands. And this time is probably the most exciting time for us because the single cell sequencing tools are exploding. And one of the main sponsors of this conference, 10X, made, for example, this type of things possible where people can take 10,000 cells, 1 million of cells, and sequence each cell individually, now with basically commercial equipment. And this is fantastic because on the experiment on the right is actually a mouse embryo that was dissociated, and each individual cell was analyzed. And you get these beautiful kind of artsy pictures. These are the UMAPs. We saw a few of these UMAPs come by in other talks. But it consists of single dots, and each dot is an individual cell. And what you really have to remember from these kind of ways to present data is if two dots are very close to each other, it means that those two cells are very similar in terms of the transcriptome, or in this case, DNA accessibility. You shouldn't interpret or over-interpret these things too much. As you say, well, this island there is a little bit more narrow than the other one, means that they're more or less variable. You shouldn't do that, but it's a really nice way to present data. And it looks also very nice. But what you can see here is that it is completely unbiased. So they take 100,000 cells from the mouse, they sequence it, and after the fact, you can analyze which genes are on and off, and you can start to identify all the cell types in vivo. So that is it's a great way to do it. But if you go back to the central dogma, that's only the first part, right? So what I want to talk today is what we're trying to do post-transcriptionally. So there's a lot of things we can do in single cells with DNA sequencing and with RNA sequencing, but there's not so much available right now to look at the process of translation. How do you turn an mRNA into a protein? Now, ultimately, I would love to do single cell proteomics, and some people are brave enough to do it, but it's still very difficult. It's the problem that you cannot really do a PCR for proteins or peptides, right? This doesn't exist. You buy a primer and then amplify a protein. It just, if you find it, you will get the Nobel Prize. But it's currently not known. And great thing about RNA DNA is that you can amplify it. So even if you start with a little bit, you can turn it into a lot. And that's good because then you can sequence it. Yeah, so basically the main technology, the main concept that we're going to use is an old concept. It's called ribosome profiling. Nick Ingolia and Jonathan Weissman, more than 10 years ago, realized that when you take the RNA that is still associated with ribosomes, and you park or you fix those ribosomes on the RNA, and then you digest away all the RNA which is not protected by the ribosome, you basically know where the ribosomes were at the moment that you fixed the cell. So this is actually quite nice. It's almost, it's as close as you can get to a translational measurement. Of course, you're not completely sure if they're moving or not, but at least you know that there's a ribosome on the RNA. And they use RNases for that. In this case, RNase I is one of the most famous ones. And we knew this, and we also knew that when you look at the protocols, you typically need 100,000 or million cells to do this type of assays. So I had a very brave postdoc. I'll show you in a second. They said, okay, let's try to get this to the single cell level. But let me give you a little bit more information on the ribosomal profiling, although we saw a lot. What to expect, right? So if you do a ribosomal profiling experiment, you get reads that were bound by the ribosome. You map them to the transcriptome. And if you look at the meta-gene plot, which is like here, right, where all the genes are basically overlaid, you look at the UTRs and the coding sequence, then of course you want to see that most of the reads will map to the coding sequence, because that's where most of the ribosomes hang out, not so much in the UTRs. And what is also very nice, if you zoom in the coding sequence, there's this beautiful periodicity, the trinucleotide periodicity in ribosome profiling. This had to do with the fact that the ribosome doesn't move like a train at constant velocity, but it moves and stops, then all the biochemistry happens, and then makes a step of 3 nucleotides, and it repeats the whole thing. So it basically spends more time in the right frame, and then you will see more of those reads. And that is a really important QC measure for ribosome profiling. Now here's Mike. Mike, again, was very brave, because he said, okay, I'm going to try to get this to work in single cells. He has a beautiful Dutch name, but he's born and raised in Vancouver, so somehow he's from an old Dutch immigrant family. But he likes the Netherlands, which I'm very happy with. So what we do typically with single cell sequencing is we first take a sorter machine, and we put each cell in an individual container, for example, in a little well of a 384 well plate. And we lyse the cells, and then we basically do conceptually exactly the same as Jonathan Weissman was doing. We have a few things that are different. So we love MNase. It's a different nuclease. It also digests RNA. What's great about MNase is you can switch it on and off by adding calcium or chelating the calcium away. So if we want to digest 5 minutes for each cell, do 5 minutes, 6 minutes, 6.5 minutes, it's super controlled. It's a little bit more difficult with the other RNases. And we, of course, have 384 experiments in parallel, and we want it to be as homogeneous as possible. So MNase helps a lot, because we can switch it on and off in a very controlled way. But in the end, it's doing the same thing. It's eating up the RNA, which is not protected by the ribosome. So in the end, what we're trying to do is to get the RNA, which is protected by the ribosome, which is the footprints. We put adapters on it. We do direct ligation to the RNA. And then with PCR, we turn it into quantities that you can use on a sequencer. Then we pool all the cells together. Each cell has a barcode. And we actually run a gel. And we cut out a band around 35 nucleotides, which is where you expect the footprint of the ribosome to be. So it's size selected. Now this is the data that you get out of this. So I'm going to show you a lot of heat maps. I'm sorry about it. There's no other way I can present data. Otherwise I have to show you one cell at a time. So I'm going to try to explain every time what's plotted on the axis. So each row in this heat map is one cell. So these are 1,000 cells. These are RP1 cells, human cell line. Here are 293 cells, 500 of them. And then each column is a position in the meta gene, right? So I aligned all the genes with the start codon, and on the other side on the stop codon. So here's the 5' UTR. There's the 3' UTR. And here the 0 is where the start codon is, and the 0 on the other side is where the stop codon is. Now the first thing you see is that there's really dark blue areas on the left and the right, which is the UTRs. You don't want too many ribosomes there. That makes sense. An ordinary RNA-seq experiment would see a lot of reads there, but we don't see it, so that's good. But the thing we got really excited about are these vertical stripes, right? And these are 3 nucleotides apart. So this is this shows that we can really measure translation. We did a lot of other controls to really show that we measure translation. This is published, so I'm going to be very short here. But this is we got really excited when we saw this. Now you can do this in single cells, but also if your gene is expressed high enough, you can even do it at a single gene level. So again, each cell here is a row. Each position is now a position in a gene, and this is a histone gene. So we're now looking along the coding sequence of the histone gene, and these are cells. Some cells were starved in arginine, so we grew them in all the amino acids, but we took out arginine, and those are the blue cells. You see these blue cells on the right. And then there was also cells that were starved in leucine. Those are the green ones. And now when you look, you see actually this group of cells here. These are all cells from the blue class, so they were starved in arginine. I see a pile up of reads at that position. And it's now in a single gene, in a single cell, right? So, and what turns out, if you now zoom in on that sequence, is there are 3 codons that are coding for arginine. And they're starved in arginine. So they have a really hard time getting through that position because they're starved for arginine. So they really slow down the translation there. And that's not visible in the cells that are in leucine, right? So I just want to show you this, that it's not only that you have to average overall genes, but you can actually go to individual genes and look where are most of the ribosomes on the gene. Yeah, so now I want to actually, what I'm going to show you is almost all unpublished. So any constructive feedback I will be very grateful for, but we still have to write the papers. So one thing we cannot measure with this technique that I showed you is if a gene, suppose a particular gene, you want to know if it's translationally controlled or transcriptionally controlled or both. So there are 2 conditions here, or like with healthy, diseased, or with salt or without salt. And let's look at 2 extremes. So one extreme would be that it's only transcription. So I have 2 transcripts, that gap DH, and I make 4 transcripts. But the number of ribosomes per transcript is the same. So this would basically be 100% transcription. This would be the opposite. I keep 2 transcripts, but I put more ribosomes on it. In the end, probably, I get the same number of proteins made, but this is completely controlled at the translational level now. Now, if I measure, what I measure is the number of ribosome-protected fragments. Well, that's exactly 4 in both cases, right? So it's completely insensitive. This assay by itself cannot distinguish. What I need to do is I need to figure out how many ribosomes are per transcript. In this case, it will be 1, here will be 2. We call this the translation efficiency. I'm going to call this TE throughout the talk. This will be great if we can measure this. And I'm going to propose 2 ways to do that. One is really inspired by the bulk kind of approaches, where you basically take an RNA-seq experiment and a ribo-seq experiment, and you compare them to each other. Now, of course, we have to be careful because we have single cells, so we have to make sure that we compare the right cells to each other, but it's basically what the bulk people do as well. This is a new idea, which I would love to get your input on, where you actually don't need RNA-seq data, but you would do a ribo-seq experiment at different concentrations of MNAs. And I'm going to try to convince you that that's enough to determine translation efficiency. And that I actually like this better because the ribo-seq technology is really different than the RNA-seq technology. It's different library preps, you use different enzymes, where here you only change one thing. It's the concentration of MNA. So its systematic errors are much smaller, I think, but it's early days. But let's see. So let me first start with the first one. So what we do is we basically have cells. We split them in 2 parts, two tubes. One tube goes for a single-cell RNA-seq experiment. The other tube goes for a single-cell ribo-seq experiment. Now, suppose there are 3 cell types in there. I will get these 3 clouds in the UMAP space. Now, the trick here is what I can do is I can integrate those 2 data sets computationally. Now, this is a little bit of magic, but basically what it means is that you ignore genes that are not changing a lot translationally, right? So you can try to figure out, I'm going to throw out genes out of this data set that are changing a lot. Because the 3 is both RNA measurements, right? So there is actually a lot of shared information. So you can do this. People use this batch correction in our field. There are a lot of different flavors, but it works quite well. I will show you. And what I can do now is I can, per cluster, I can start to compare the RNA-seq cells to the ribo-seq cells. But I do this based on each cluster separately. And then, for example, what I would see is that comparing the RNA to the ribosome protected fragments, in cluster 1 it might go up. Now, that doesn't mean anything because, as I said, it's different technologies. It's apples and pears and different units. But if I see, for example, a cluster where cluster 2 and 3 have the same level of RNA, but in one case the RPFs go up, the other one go down, then something interesting is happening. So in this you can mathematically do this really robustly. I won't bore you with details, but this is basically what you're going for. It is very similar how people do it with bulk data, but then of course then you don't know which clusters there are, but it's very similar. Now, I want to do a little intermezzo here, because if you think about the RPFs, the ribosome protected fragments, they are sampled from the whole transcript. And most RNA-seq technology, including 10x, right, is from the 3' end. It's only sequencing the last little piece of the RNA, which is next to the poly-A tail. So if I want to compare those data to our data, it's not really fair because I'm sampling from the full transcript. And also, many mRNAs that are interesting, including the histone gene I showed you, they don't even have a poly-A tail, so I will never see it with RNA-seq. So we need to, as a reference, we need a full-length transcriptome measurement, ideally for genes that don't require poly-A tail. So how to do that? How can you detect the transcriptome in a single cell, even if it's not polyadenylated? Why don't you add a polyadenylation tail yourself? Let's do it artificially. So suppose in this example, there's one RNA which doesn't have poly-A tail, the other one has. Typically happens in these experiments is that it gets fragmented. Either you do this on purpose or you can't avoid it. Then, in our case, we can just put a poly-A tail on all those fragments. And this is, of course, that we use an enzyme from E. coli called poly-A polymerase and just put an artificial tail on all these molecules. And now you can use all the existing technology that's already out there to detect all these molecules. And this is in contrast, what I was saying, to the 10x system, which is great, but it only will sequence that little red piece there. That's the only one that will survive in the 10x system. CellSeq from Itai Yanai, actually, he developed it. Great system. It's the same thing. You only look at the 3' end. And this also has consequences for the sensitivity, right? Because this gene, this red gene here, I have 4 possibilities to see it, whereas if you have only the last part, you only see it once. So by doing this trick, your sensitivity also goes up. But at the same time, you also will see all non-polyadenylated RNA, and you will see the full structure, including all the splice variants, right? So that's quite nice. So we call this vast transcriptome analysis of single cells by A-tailing, Vasa-seq. Now this is not completely random. This is King Vasa. Gustav Vasa is one of the kings of Sweden. And one of the postdocs is from Sweden. And he is very much into King Vasa because apparently King Vasa scared away all the Danish people out of Sweden. And there's somehow something going on between the Danes and the Swedes. They don't like each other. So Frederick really wanted to call it Vasa-seq. And we had a great collaboration with the Hofelder lab. There's actually a person from Belgium, Joachim. And he did a lot of the technology for the droplets. As you remember, the 10x system is a droplet-based technology, which is great. We couldn't use it because we need 2 reactions. So in the 10x system, it's a one-shot reaction. All the chemicals are in the droplet, and then you only have to basically go through a PCR cycle. But we need 2 reactions. We have the polyadenylation, and then we have the reverse transcription. So we needed somehow to have the droplets, but also be able to get another injection into the droplets later. Now that's exactly what Joachim is really good at. So they have this microfluidic system. They're in Cambridge University in UK, where you can have the droplets. They went through the first reaction. They come back through the chip, and you inject a little bit of chemical mixture again. And hopefully the video works. Yeah. So here, these are beads. Beads have all the DNA primers on it, and there's also a cell in there. They already went through the first polyadenylation step, and now they're prepared for reverse transcription. So now they get the RT mix in there. After that, they go all in the PCR machine, do the reverse transcription reaction. And then, in principle, you can keep doing this. You can do 5 steps if you want. So we're really happy with this system because it allows you to really be a little bit more creative than just one-step reaction, because not all biochemistry can be done in one step, right? So this is really nice. And this is a very slow down. So this goes like, if you do it at real time, you can't see it. So this goes very fast, and you can process 10,000, 100,000 cells. So to show this technology, how it works in vivo, we decided to look at the mouse embryogenesis. So we took mouse embryos ranging from 6.5 day to 9.5 day, dissociated them, and run them through the microfluidics and make UMAPs. Again, it's very similar. beautiful structure. I'm not going to go through. It actually takes an enormous amount of time to figure out what each cluster is, especially for a physicist like me, because I don't know all these different versions of the mesoderm. But we got a lot of help from the biologists. But one thing which is really nice and which is different from our system than from say the standard system is that we also have a lot of introns. Introns are all full length, and we detect them. And this allows you actually to predict the future a little bit, because if you see a lot of transcripts that still have introns in it, it means that they were made very recently, because these introns are removed quite quickly. But there's also RNA which hanging around has only exons. So then well, this old RNA, it was probably made a few hours ago. So based on this, you can actually replace each dot here with an arrow. Where is it going? And that's what you can see there. So you can now see this kind of winds in the UMAP, the direction of flow. And what's really nice is that we actually already knew the answer, because we have different embryonic stages, but we didn't use that information. But you can see that the arrows all go in the right direction, right? So these are red blood cells. The E6.5 is the dark color, and all the arrows, they point up, going to later stages. But the arrow is determined not based on the stage of the embryo, but based on the intron retention. So this shows that this data is also very useful for predicting the future, because of course you kill the cells in this whole process. You can never look at it again, but somehow by comparing the intron retaining genes to the rest, you have some idea of where it would have gone if it would have still be alive. Now another thing is the non-polyadenylated genes are detected. So these are all histone genes, many histone genes in the genome. Now, this particular H2B gene is only expressed in this very small island here in the UMAP, which is very early pluripotent cells. And then other H2B genes get fired up later. They're also very cell type specific. Again, you couldn't measure this with normal RNA-seq data because it doesn't detect the poly-A tail. And then, of course, another obvious application is splicing. And Guillermo here actually did a lot of the heavy lifting here, or all the heavy lifting, for our data when he was in the Hamburg's lab. So he's an example of the development of the heart, where you can see here this exon 6B is flipped for 6A. So it's enormous amount of data that really allows you to go beyond just counting transcripts. So we're super happy with this. It was published last year. Just check it out. Simple step, just add poly-A to everything, and you can use, in principle, the same technology. You will get ribosomal RNA as well. We try to deplete that, but if you like ribosomal RNA, which I do actually, you don't have to deplete it. Okay, there was an intermezzo. So I told you I would talk about translation, but this was really a transcription. But we need it as a reference, right? We need a full-length RNA-seq reference to compare to the ribo-seq data. And as I said, we're going to basically integrate the data, and this is how it looks like. So this is a human cell line, just very boring, going through the cell cycle. Actually, what we did is we sampled the cells randomly, but we also had two separate populations. One is a mitotic population. So what you can do is you can take the plate and you smash it on the table. The cells in mitosis, they detach, and you can enrich for them, because mitosis is pretty short. So if you don't do that, you will get only a few cells in mitosis. But these purple cells are shake-off cells, so they're in mitosis. And then G0 cells, which is probably not a good word, G0, but these cells are grown up to confluency. So it's the same cells that were just grown until they touch each other, and then when they touch each other, they will stop the cell cycle. So those are the guys in purple. All the blue cells are just randomly sampled from the cell cycle. Now what you can see is now after I integrate these two data sets, so I take RNA-seq and ribo-seq and you integrate them, and they beautifully give the same UMAP. And what I can do now is I can what we call define communities. So these communities are basically little circles here that have both RNA-seq cells and ribo-seq cells in it. And they live very close, in this UMAP to each other. So they're pretty similar to each other. So what I'm going to do now for each of these communities, I can calculate the translation efficiency. I cannot do it for a single cell, because in a single cell, you only have either ribo-seq or RNA-seq. But if I say, okay, I'm looking at this cell and all the neighbors that are very similar, they're basically undistinguishable. Let's compare those 20 cells. Or you can see the number of cells there. Sometimes there are 100 cells in a community, sometimes 50. But this is enough to estimate translation efficiency. Now let me give you a few examples. So this is a gene called TPT1. If you look at the RNA level, it looks like this. So you see basically a red kind of signal all over the place. So the cells go like this through the cell cycle, right? G1, S, G2. So they go clockwise through this, if they will be alive. But the transcript levels don't change much. But if you look at the RPF levels, they actually are high in mitosis and they go down in G1 and they go up again. So it seems that there is something going on. And if you now do the calculation for the translation efficiency, is what you expect, of course, is that in G1 and G0, translation efficiency much lower than in mitosis or in S phase. Now, that's one example. This is another gene, NOX1, which is, by the way, I have to tell you, PTP1, if you look at the name, it's tumor protein translationally controlled. Right? So it's not a great discovery here, but it's good to know. So NOX1 is much more unknown gene, by the way, is doing exactly the opposite. So when you look here at this transition going from S phase to G2, you would say on the RNA, I don't see anything changing, but there it basically plummets down. Now, it goes from high to low. And again, if you do the calculation, you see indeed that the translation in mitosis is repressed. Now, another heat map, where now each row is a gene. So because I'm going to make this type of plots for every gene that I can measure. And the columns are cells, which are basically ordered based on the cell cycle stage. So this was the two examples I showed you. It turns out that many genes behave exactly the same as these two examples. Now these TPT1 class, these are known genes that are translationally regulated. The ribosomal subunits are in here. And these are repressed in mitosis, or G0. So in cells that are kind of not dividing, definitely G0, they're not dividing, these are repressed. The other ones, which are very interesting to me, are repressed in mitosis, this big class here. And then there's a little bit of a more fuzzy class which might be activated in mitosis, but I'm not going to focus on that one. Now what you can do is you can take all the genes in that cluster and look for sequence motifs, like we saw in the previous talks. And this one, cluster 1, has a very strong CU enrichment. Now this is known. This is the so-called 5' terminal oligopyramid tracks, or the top genes, the 5' top genes. So this is something that we recover. It's interesting that it happens during the cell cycle, right? Well, these genes are definitely picked up if you really switch off proliferation, but even in G1 compared to mitosis or S phase, you see these ones show up. This one is more unexplored. If you look at the sequence motif, it's either a G or A. By the way, this is a function of transcription start site, so it's the distance from transcription start site. And there is some other sequence motifs later, which I will focus on in a second. But let's look at this cluster 2, because that's really more novel, I think. We did very similar thing as in the previous talk. We take the UTRs and we fold them in the computer. We look for secondary structure of these UTRs. And short story, if you look in the 3' UTR, there's no real difference between the different clusters. They basically have the same delta G here. Delta G is a measure for how stable is the secondary structure. But cluster 2, which is this mitotically repressed cluster, seems to actually have stronger secondary structure than the other ones. So then we started to look very carefully for motifs in there. So not only from the transcription start site, but actually by having a moving window moving around. Before I go there, I have to show you actually that when you look at the fraction of the reads in the UTRs, this repression in mitosis cluster is also quite special because it had a lot of, or much more reads that land in the 5' UTR. If you look in the 3' UTR, there's almost no reads there, so almost no ribosomes. But the blue cluster here, the mitotically repressed one, have more reads in the 5' UTR, suggesting perhaps that the secondary structure might obstruct ribosomes, or at least we see more. It might, of course, to be completely honest, if we see a read, we cannot prove that it was a ribosome protecting it, right? Might be an RNA binding protein, but at least we see more reads than in the other condition. And then we search with a moving window, we get a very strong motif here, which I saw in a poster yesterday as well. So we actually are discussing together. So it was in a P53 unfolded protein response kind of poster. So it's a very different context, but it's exactly the same motif. So very interesting, and we'll continue, talking to each other. But this is a very strong motif, and actually also in the previous talk, also this is a candidate for a quadruplex in the RNA. I'm not proving it's there, but there's definitely a motif that's overrepresented, and perhaps it's related to this obstruction of the ribosomes moving to the open reading frame from the 5' UTR. Okay. Now I want to show you the other method that we think of to use to calculate the translation efficiency. And again, this is a method that will not require any RNA-seq, so this might actually also be useful for bulk ribosome profiling. And this actually goes back a long way, actually before I was born, which I'm happy to say, I'm also getting. So there's a paper about MNase, and MNase actually has two phases. It is really mostly an endonuclease, which is basically a scissor, so it cuts somewhere. But if you wait long enough, or if you have high concentrations, it's an exonuclease. Like I look at this as a Pac-Man that will bind to the end of the molecule and eats it up along the molecule. So not like a scissor, but like a Pac-Man. So we use it. So if we would do very low concentrations, we would randomly, let me show you what I mean by this. So a very low concentration MNase, my assumption is that not the whole transcript is covered in ribosomes. So I'm assuming that at most 30% of the transcript is covered in ribosomes for very highly translated genes, but most of the RNA is actually accessible for MNase. So if you just have a little bit, you would just randomly cut it, and in this particular example, what you see is that you actually will get a fragment, but it's not protected by ribosome. This will make it through the size selection, and I'm going to call this a UPF, an unprotected fragment. If you go to higher concentrations, the MNase will start to cut like an endo, but also will start to eat up things. And what you will get, sometimes you will still, one of the unprotected fragments will go through, will make it through, but definitely at high concentration, everything is eaten up until, really the boundary of the ribosome is completely nice and tight. And that's what you want for ribosome protected fragments. This is where we typically do the experiment. But we started to do titrations, and we saw there's actually quite interesting stuff happening here as well. If you look at the length distribution, so this is, again, a heat map, where this is now the distribution of lengths. So it's a histogram. Each column is a single cell. So in one cell, I look what are the lengths of the fragments. And you see that normally we detect everything for low concentrations between 25 and 45. This is really how Mike cuts the gel, basically. But it's quite uniform. So this doesn't look like ribosome protected fragments. No, these are just random fragments that just randomly make it through the size selection. They were cut randomly by the MNase, and they have the right size. If you increase now the MNase concentration, you start to see at very high concentration a beautiful, really sharp distribution. This is what you want to see. This is the ribosome size. And here you see the exonuclease activity. Here you have actually quite long, but they're chopped until you really reach the boundary of the ribosome. So there's quite a nice transition. So how we interpret this is, if you here, you're really doing transcriptomics. And here you're doing riboseq. So why not just compare those two concentrations to each other? And what I love about it is just one thing that you change, right? Rather than I have another kit, another polymerase, this is just one thing you change. So systematic problems are not such an issue. Now, we'll just give you one example of a gene. Again, so what we do is we look at this, how many fragments do we see for this particular gene as function of MNase. What you actually see is that you get more fragments at low concentration than at high fragments. And this is because the unprotected fragments are also there. And this is in mitosis. So this is one of these genes that is translationally repressed. So after you add a lot of MNase, there's almost nothing left. So this means there's almost no ribosomes on this transcript. But when you go interphase, it looks like this. So these actually are almost constant, first approximation. So there are ribosomes there. But if I now compare the different concentrations, I would say, well, at low concentration, I'm mostly looking at transcripts. and they're very similar. And at high concentrations, I look only at the ribosomes, and they're gone in mitosis, but they're still there in interphase. So from this, I can basically get a ratio of these two things. If I take a ratio of ratios, so the ratio of these two divided by the ratio of these two, I can get an estimate for translation efficiency, which I can compare to my previous method, and they correlate quite well. And this, this different, completely different methods, right? So this, actually, I got this plot 2 days ago myself. I made it. So I still have I need Mike's approval, but random positive correlations for 1,000 genes, they typically don't show up like this. But this is the disclaimer, and Mike did not double check this. But there's a Pearson correlation of 70%, which is really nice. And again, this might actually be useful for the bulk ribosome profiling people, because this doesn't require single cells. Yeah, so let me finalize the RiboSeq part. We developed this technology, and now we're very much into figuring out if cells regulate translation, transcriptionally controlled, which you can do now in two different ways. They are pretty much give the same number. Really want to get to the real numbers and say, okay, it's 30% of the transcript is covered in ribosome. There we're not there yet, but I think we will be there in half a year or so. And we have this interesting biological observation is that in mitosis, there's repression of a subset of genes. They have this beautiful GGC repeat. There is also secondary structure involved. If you have any good ideas, let me know. Again, realize I'm a physicist, right? So I need help with the heavy-duty biology here. But I'm very, very happy. I saw this poster yesterday, which basically had a very similar motif. So hopefully we can help each other there. Now, the last few minutes, I want to talk about also something I never presented before. And this is about tRNAs. So it turns out that if you look at the RiboSeq data, it's in our supplementary figure, but we didn't put it as figure 1. Almost 60% of our reads are tRNA reads. What we first did is we just threw them away until I thought, wow, tRNAs are also important for translation, right? They really translate the sequence in the mRNA to the polypeptide. So perhaps we can do something with these tRNAs. And we had also all these titration experiments that I already showed you. And I thought, perhaps we can do something with this. Now, tRNAs are very interesting, super important. They're heavily modified. So the RNA is heavily modified with many, many different options. Mostly in the anticodonal loop. So this point here, the red residues, they're in contact with the mRNA. This is where the decoding happens. And they're full with RNA modifications. So we saw this. Okay, endogenously, what happens is that tRNAs also get digested. And this was in a review I found, is that they so in the cell itself, right? So tRNAs are chopped into pieces, and it says emerging data suggests that this fragmentation might depend on the modifications, and these modifications might protect or actually do the opposite, make it more likely that there is fragmentation by endogenous RNases. So we thought perhaps we can do a external digestion with our MNAs and see if the MNase digestion is also changing the digestion of these tRNA fragments. There's actually a project by a master student who I mentored myself since a long time. I'm not the director anymore, so I have a little more time. So I started to work directly with a master student, and he's luckily now a PhD student in the lab, really good. And this is the type of data, this is experimental data that basically is a side product of all the ribosome profiling we did. Again, we do a titration experiment. So there's an MNase concentration here are the different. It's increasing from the top to the bottom. And this is one example tRNA. So this is a tRNA, it's an arginine tRNA, 75, 76 nucleotides long. And what I'm showing you here is where the 3' ends of the tRNA land. Right? So what you see is there's something happening here, and then something happening in there. Well, of course, this is the end of the molecule. That's where the 3' end is. But this is interesting because it's actually cutting the tRNA in half. So our MNase, not endogenous, because the top row is when there's no MNase. It's completely dark. So there's not so much endogenous digestion going on, but we digest it in the lysate. And what you see is that halfway the tRNA, there's a lot of cutting, or there's a lot of cutting going on. Here's where the molecules stop. But also when you increase the concentration aminase, you see the exonuclease activity happening here. So for low concentrations, it stops here, but then the Pac-Man eats his way to the right, left, when I increase the concentration aminase. And the same thing happens on the other side. So we think about it as, you start with a full-length tRNA. At some concentration, you chop it into two. Right? That is the endonuclease activity. You just, with the scissor, you cut it in two. And then, at higher concentration, the exonuclease activity activates and starts to eat it up from the right to the left. And it becomes shorter and shorter as the Pac-Man goes through the tRNA. Now, my physicist background can't resist. These are chemical equations. There's a cascade of chemical equations, right? There's basically a cascade. You can turn this into differential equations, which you can actually solve completely analytically, which means you don't have to do any computer simulations. If you take like SymPy, for example, you can solve this. It's the solutions are not pretty. If you would need, a whole slide to show you the result, but it doesn't matter. It can be solved. So now what we can do is we can compare our experimental data to this kind of digestion models. Again, the same data. What I'm going to do now is I'm going to plot the data, 90 degrees. So I'm going to plot it as function of aminase concentration. Right? So you get this type of curves. This is on the left, this is on the right. And now I'm going to try to fit the model to it. And it does a great job. So each curve here is basically one fit parameter because it tells you how fast the Pac-Man is going. So in this case, I have 6 fit parameters. But that's okay because I have 6 curves. Right? You always have to be super careful with fitting things, right? Physicist says, you can fit everything. You fit an elephant with 4 parameters, and the 5th parameter you can make the tail wiggle. So. But if you think about a straight line, you already need 2 parameters, right? So I have peaks here. So for something going up, going down, you need at least 3 parameters, but I'm only using 1 parameter. So this shows that this is not overfitting. This model is probably pretty reasonable. I mean, it's never perfect, but as you can see, it is quite nice. So now what happens is this whole heat map gets reduced to 4 or 5 numbers, which are rate constants, telling me how fast do you eat through the tRNA. And now what I can do is I can compare different conditions. Right? Now first I have to do a positive control. I want to make sure that this really works. So there's a very nice RNA modification which we call Q. It's a very complicated name, but it's based on quinine. So quinine is a small molecule which is actually, we cannot be made by the human body. So you have to take it in through the diet. And so we use that fact. So you grow human cell line in FBS. So FBS contains quinine. So what you do, you dialyze the media. And now you lose all the quinine. And then you buy quinine externally and you add it. So I have now a control where I'm pretty sure there's no quinine. So this modification, the Q modification, cannot be made. And I have a control where I externally supplied it. So in the left case, I have only tRNAs without the modification. And with the quinine, it should be there. Now the modification is very well defined. It's on the wobble position. It has the G is replaced, biochemically turned into the Q. So I know exactly where it should be. And I also know there's only 4 tRNAs that should do this. So if you remember the codon table, these are the 4 codons that are possibly have the Q modification. These are so-called GUN tRNAs, so G-U-N. Right? So of course here you have to invert it, to reverse complement it. But these are the 4 that you would expect to have the Q modification. And, it's important for the wobble position so that actually these tRNAs can also encode the other codons. Now here's my volcano plot. Right? I couldn't leave it behind. So what you see here is the fold change, the digestion rate. So I'm plotting here, each point is a tRNA, but it could also be one of the rate constants. So there's, you see one tRNA many times because there are 5 rate constants for the tRNA, right? Depending on where it is. And I'm comparing minus Q to plus Q. And this is if you're on this side, it means that the digestion is faster in the absence of quinine than with quinine. Now, what you can see, is you see almost only guns here. So GTC, these are almost only guns. And this means that this idea we have that this tRNA digestion is dependent on the modification actually works. You can identify exactly the tRNAs, only those 4 tRNAs that have the modification. Another way to show it is in this heat map, where now each row is a tRNA, and each column is a position in the tRNA. And what you have to look for is really red or really blue pixels in this heat map, because they show that there is a lot of change in the digestion rate at that position of that particular tRNA. Now, there's a really big red dot there, and this is exactly at the gun here, the GTC, and it's also exactly at the right position. Right? So it's position 33. That's why we see a huge difference between digestion. So I think this experiment shows this method works. You can identify with single nucleotide precision in a tRNA where the modification is. Now, this is very early work, and we're happy that this kind of positive control works. So to show you a little bit, if you just now go through the cell cycle, now you compare G0 confluent cells to cycling cells, we also see a few red pixels and blue pixels. We have no idea if these are real positives yet, but just to show you that, for example, if comparing interphase cells to non-interphase cells, we see this very blue pixel, which is exactly at the pseudouridine site. So suggesting that perhaps, and we have to validate this, but that the pseudouridine actually might be modulated during the cell cycle. And the same here. They have also, there's positions which are not known to be modified, so I'm also not sure what to make of it, but this is something that we really want to explore further. And again, as I said, it's a master project, but I'm very happy with it because I mentored the master student myself, so I wanted to show it to you. But I think there's something in here that we can do, and this is all single cell resolved, and tRNA in single cells have not been published before, so I think this might be useful. Most people say these tRNAs are not changing, but I'm not sure yet. Yeah, of course, where we're going is to go to even more complex systems, right? So we already have data sets that we're chewing on, where we have a mouse intestine with many different cell types, or organoids, or mouse embryos. That's, of course, the beauty of this technology is that it works for, as Itai and I said, any cell. Doesn't matter if it's a mouse or a zebrafish. So, of course, these data are much more difficult to analyze because you have many more clusters, but we're doing our best. And yeah, I just want to thank the lab because, in particular Mike, who came out of Canada and got a single-cell ribosome providing the work, which was a big project. It, of course, didn't work immediately. It took forever to get working, but then when it worked, it really worked every day. And yeah, it's the same for the whole lab. We do typically high-risk projects, and they don't give up. So it's often that I say, well, shall we stop? And they say, no, let's try again. And they're often right, then it actually works. So I'm super happy with this attitude. And I also, of course, want to thank the funders that pay for the bills. Yeah, thank you very much.